Hello everyone, welcome to day 2 of genetics. Today we are going to see mutations. Remember, mutations are nothing else but some permanent change in the DNA. They may affect the germ cell, they give rise to inherited diseases. Remember, the gene mutations can be classified into number 1, some point mutations which occurs within coding sequences. This point mutation refers to, that means a single base pair will be substituted with a different base. Suppose if these mutations alter the meaning of the sequence, we call them as missense mutations. Missense mutations may be either conservative and non-conservative. Suppose if this missense mutation causes a little change in the function of the protein, we call it as conservative missense, and if there is a complete function is altered, we call it as non-conservative. Best and excellent example for missense non-conservative mutation is sickle cell anemia, in which in the beta globin chain of the hemoglobin, GAG sequence in mRNA which codes for glutamic acid is replaced by GUG code in mRNA which codes for valine, leading to some alterations in the physicochemical properties of the RBC, leading to sickle cell anemia. Nonsense mutation. Suppose if this base pair or this codon is substituted by a terminator codon or a stop codon, we call it as nonsense mutation. Best example, beta thalassemia, in which in beta globin chain the glutamine, that is CAG, that sequence is replaced by UAG, which is stop codon, so it will cause termination of protein synthesis. No beta globin change will be produced, causing beta thalassemia. Second class, mutations within non-coding sequences. That means there will be some alterations in the promoter enhancer sequence causing marked reduction in the total marked reduction, as well as there will be total lack of transcription. This is also classically seen in thalassemias. Then deletions and insertion. Remember 2 points. If this number of base pairs that we are either we are adding or deleting, if it is in the multiple of 3 or 3, the reading frame will remain intact. Fine. So the abnormal protein, either it will be absent or sometimes that protein will be produced in excess. At the same time, if the number of affected coding bases, it is not a multiple of 3, there will be a complete alteration of the reading frame of the DNA strand, and that is what we call it as frame shift mutation. The best example we have is 3 base deletion in the common cystic fibrosis gene. Okay, so that there will be a synthesis of a protein which lacks the amino acid 508, that is phenylalanine. Okay, yes, and remember here it is a multiple of 3, so cystic fibrosis, it is not an example of frame shift mutation. Then coming into some other alterations, that is translocations, Philadelphia chromosome, which we will deal in later, that is translocation between BCR ABL genes, chromosome number 9 and 22, leading to chronic myeloid leukemia. The next class, alterations in the non-coding RNAs, okay, which will, that is miRNA, long non-coding RNAs, and their microRNAs that also produce some features. The diseases which because of these mutations are not that much well established. We have trinucleotide repeat mutation, that is a special category of genetic anomaly. Here actually there will be some thousands or hundreds of repeats of a particular sequence. For example, we have Fragile X syndrome, in which there is a gene called familial mental retardation gene, FMR1, and that sequence is CGG. This sequence CGG is repeated some 4000 times, and as a result what will happen, the normal gene can't express properly, which leads to intellectual disability, that is Fragile X syndrome. Okay, yes, and remember the characteristic and distinguishing feature of trinucleotide repeat mutation is they are dynamic. That means the degree of amplification increases during gametogenesis, that is dynamic. The word dynamic means the degree of amplification increases during gametogenesis. Fine. See, here we have a picture of single base deletion. Okay, see, whenever there is a single base deletion at the ABO blood group, what will happen? Single base. It is not a multiple of 3. So what is the type of mutation? Very good. It is frame shift mutation. See, there will be premature. See, CAG. UG. So, okay, so normal glutamine replaced by stop codon. This is nothing else but beta thalassemia. Okay, beta thalassemia. So this is again example of what? Nonsense. It is nonsense point mutation. Best example, beta thalassemia. So I hope you have enjoyed the session. Thank you. And in the next session, we will start with the different patterns of Mendelian inheritance. Thank you.